And welcome again to another video in our Biology 2. And after finishing up with the proteins last video, we'll now proceed with another important topic, and that is the oxyribonucleic acid, or DNA. And we're done with the four biomolecules with this. So we started off with the carbohydrates and the lipids, and then proceeded with the proteins, and now we finish off with DNA, or the deoxyribonucleic acid. So for this module, which is SLG 2.1, we're going to be talking about the roles and components of the nucleic acid, which brings us to the objective that for this module, we want to determine the roles, the functions of nucleic acid in connection to its structure. So this will imply to us that we need to be able not only to memorize, recall the different structures, different important parts of the nucleic acids, but we must also know what would be the roles and functions related with these structures. So we start off with this question. What is the function of the DNA? Now, the function of the DNA is to store, transmit, and help express hereditary information. And we talk about hereditary information, we're also talking about the final form, the final expression, which would be the phenotype. So everything about us, everything in living organisms, as you can see, would be related or would be brought about by the DNA. So the color of our hair, the skin tone that we have, the texture of our nails, our hair, the colors of our eyes, all of these phenotypes, all of these traits are expressed or are fundamentally related or produced by what we call as the DNA. So again, the function of the DNA is to store, transmit, and help express hereditary information. So with that, let's look at some important things about the nucleic acid. So as we mentioned already, it would be transmitting, storing, and helping express hereditary information, and that the amino acid sequence of a polypeptide is essentially programmed by a unit of inheritance called the gene. So, as you already know, that the polypeptide or the protein would eventually be coming or would eventually be produced from the DNA. So, for example, we have the melanin. The melanin produces skin color on our skin, whether it's darkish or whether we are light colored in our skin. That melanin was produced from an RNA, and the RNA eventually was produced from a DNA. And that particular section of DNA that would be coding for a polypeptide is called a gene. Another thing here is that the gene consists of the DNA, a nucleic acid made of monomers called nucleotides. So please take note that there are very important terms here like the word nucleotide. So if the monomer of polysaccharides will be monosaccharides, if the monomer of proteins would be amino acid, the monomer of nucleic acids will be called nucleotides. We'll be looking at that a little bit later. So first of all, let's talk about the history of the discovery that the DNA is indeed the genetic material, because from the time of Mendel, where Mendel was able to elucidate, able to discover the ratios and the patterns of inheritance. So we're familiar with 1:2:1 ratio for the genotypic ratio, and 3:1 ratio for the phenotypic ratio for the complete dominance. From then on, the next question scientists had was, what is the physical basis of this inheritance? Because Mendel was able to determine this, but he was only thinking about theoretical units of heredity because he hasn't even seen it, but he was able to deduce it, showing how a genius indeed that Mendel was. But after Mendel and discovering those patterns of inheritance, scientists continue to discover, scientists try to discover what would be the physical basis of this particular inheritance. Later on, scientists were able to determine that it's found in the chromosomes, and the chromosomes would have a lot of macromolecules present. So eventually, at the end, they were able to discover that the DNA is indeed the genetic material. So we'll be looking at the history, but we'll only talk about 3 of these researches because we don't have enough time to talk about all of the researches. First of all, we have here Griffith's experiment, and this is very interesting. First of all, for Griffith's experiment, he made use of 2 kinds of bacteria. The first one is a living S cell, and the reason it is called S cells because, as you can see in the picture, the cells or this bacteria produces a very smooth colony around itself, and one of the important characteristics is that it has pathogenic capabilities. It's able to produce a disease and able to kill organisms. So that would be the first kind of cells that he or bacteria that he made use of. These are living S cells. The second thing he made use of would be living R cells, and the reason they are called R cells because they don't produce smooth colonies, but they produce rough colonies. So the outside or the outline of the colonies produced are very rough. Now, the characteristic of the living R cells is this non-pathogenic. So even if you infect organisms with this, they will not die because they do not produce any kind of disease. Okay, so we have those two, the S cells which would be pathogenic and the R cells which are non-pathogenic. So in his experiment, he first made use of the living S cells, placed them inside of a syringe, and then he injected it to living mice. And what's the result? Well, expectedly, since our S cells are pathogenic, the mouse or the mice died because of the development of diseases. So this is actually expected. The next part of his experiment, he made use of living R cells. These are non-pathogenic. And he placed this inside of the syringe and then he injected this into mice. So what would be the expected outcome? Since they are non-pathogenic, it is expected that the mouse will not die, and that actually what happened in his experiment. The mouse remained healthy. So in this case, the S cells causes the mouse to die because of the presence of diseases, while the living R cells, non-pathogenic, resulted in healthy mice because they did not die. Now, the third part of his experiment wherein he killed the S cells. He placed it into a hot bath and then essentially killed the S cells. Now, since the S cells, although they were used to be pathogenic, they were killed, it is expected that they are non-pathogenic, which means they are not able to produce a disease because in the first place they were killed using heat. So Griffith placed it inside of the syringe, injected into the mouse, and then what would happen? Expectedly, it would not die, or the mouse will not die because the S cells are killed in the first place, and that was what actually happened in his experiment. Now, what is very interesting is the last part of his experiment, wherein he mixed heat-killed S cells and living R cells. So this means that the S cells are not living anymore. Means they're dead. They are no longer pathogenic. They are not able to produce disease because they were killed in the first place. On the other hand, the R cells are living, which means although they're living, they will not be able to produce any kind of disease because in the first place, R cells are non-pathogenic. So what would be the expectation? The expectation would be, since the S cells are dead and the R cells, which are non-pathogenic, are living, even if you inject this inside of the mice or the mouse will remain healthy since the S cells are dead and the only bacteria injected are non-pathogenic R cells. But what is interesting was, after injecting, the mouse died. And when they took out the blood and cultured it, they found living S cells. So the question, where did the living S cells came from? In the first place, you injected dead S cells, and then you injected with it living R cells, but suddenly the living S cells are found in the blood of the mouse, causing the mouse to die. And the conclusion of Griffith's team is that somehow the R cells were quote-unquote transformed from non-pathogenic becoming pathogenic. From living R cells, they were transformed into living S cells. So in other words, there is something that is inside of the S cells, the heat-killed or the dead S cells, that were able to transform the living R cells, causing the production of living S cells inside of the mice, causing the mice to die. So this is now a showcase that there is indeed a transforming principle that would be producing phenotypes, or would be producing characteristics or trait. So that is what they were trying to look for later on in the next experiments or researches that was done by scientists. They already know that there is a physical something that produces characteristic. There's a physical something that would be producing traits and phenotypes, and this will be related with what Mendel said as the inheritance factors. So at this point, they know already that there is this transforming principle. the next experiment that actually showed us what is that transforming principle is Avery-MacCarty and MacLeod's experiment. Now, at this point in time, they already know, scientists already know that lipids and carbohydrates are not the transforming principle, and they're left with these three biomolecules, which would be the proteins, the RNA, and the DNA. So among these three lies the transforming principle, and at this point they still do not know. So this experiment or this research actually led them to discover that the identity of this biomolecule which would be producing, or what we call as a transforming principle. So in their experiment, they made use of heat-killed S cells, and then using that, they remove the lipids and remove the carbohydrates. So what's remaining inside of this test tube after blendering it, after removing the lipids and carbohydrates, would be the presence only of the proteins, the RNA, and also the DNA. Now in this case, they made three of these setups wherein they will be removing one of this biomolecule from each of these test tubes or each of these setups. So for the first, they would be removing the protein, which means the RNA and the DNA will be remaining. The other one, they will be removing the RNA, which means that the proteins and the DNA will be remaining. While this one, the third one, they will be removing the DNA, which will, proteins and the RNA will be remaining here. The idea here is that if the setup still produces the S cells, or we're able to transform, then the biomolecule that you remove is not the transforming principle. But once the setup did not produce any S cells, you would be expecting that what was the biomolecule that was eventually removed in the first place is the transforming principle. So looking at this one, in the first setup, they added proteinase, so which destroyed all the proteins, and so what is remaining is only the RNA and the DNA. So if the protein is the transforming principle and it was removed, you would be expecting that there will be no S cells produced, or there will be no transformation occurring. But the setup showed that S cells were produced, living R cells were transformed by the heat-killed S cells. So this means even in the absence of the protein, the S cells were still produced, the R cells were still transformed, which means the protein is not the transforming principle. In the second setup, they added ribonuclease and the RNA were destroyed, which means if the RNA, if the setup showed that there is no S cells produced, then the RNA is the transforming principle because it was destroyed in the first place. So after removing the RNA with ribonuclease, what remains are the proteins and the DNA, and then they added R cells, and what resulted? They found living S cells, or S cells appeared. So this means that the RNA is not the transforming principle, because even in the absence of the RNA, there is still the production of S cells. So something still transformed the R cells into S cells. Finally, in the third setup, they made use of the deoxyribonuclease, so there is the destruction of DNA. So no more DNA is present. The only thing present here is the RNA and also the proteins. So what happened here is that when they removed the DNA and they added the R cells, lo and behold, there are no S cells produced. So this gives us the conclusion: in the absence of the DNA, there's no transformation that happens. There are no S cells produced from the R cells. This concludes, or leads us to the conclusion, that the DNA is indeed the transforming principle, because in its absence, no transformation happens. In its presence, transformation indeed happens. And then finally, Hershey-Chase experiment was the one that were able to really solidify that the DNA is a genetic material. In this case, Hershey and Chase made use of radioactive elements. So for example, for this one, they made use of radioactive sulfur, which can only be found in proteins. On the other hand, they made use of radioactive phosphorus, which can only be found in the DNA. So the idea here is that when they infect bacteria with radioactive labeled bacteriophages or viruses, they already know that the viruses are actually produced or replicated inside of the bacteria. So there is a set of instructions, the genetic material that produces the other viruses. So they have to make sure, is it the protein that would be the one that would produce more viruses, or is it the DNA that produces more of the viruses inside of the bacteria? So their idea is whatever goes inside of the bacteria is the one that would be the genetic material that produces the viruses. So in the first part of the setup, they made use of radioactive labeled sulfur, which means that the protein coats around the bacteriophage would be the one that would be labeled. And they allowed the bacteriophage to infect the bacteria over here, and then after that they agitated this one using a blender to remove the protein coat from the bacteria, and then after that they centrifuge that, and then they try to determine where is the radioactivity found. So after centrifuging it, they found out that the supernatant or the liquid contains radioactive elements, which means that the protein coat of the viruses remained outside and they did not go inside of the bacteria. So this means it's not the one that would be the one that will be producing or giving the instruction to produce the virus, because in the first place they never entered the bacteria in order to produce more viruses. But this was later solidified with the second part of the experiment, wherein in this case they made use of radioactive labeled DNA, and then allowed it to infect the bacteria. So when they blended it to remove the protein coat from the bacteria, and then they centrifuge it, they noticed that the pellet, which will containing all of the bacteria, and the liquid, which will containing all of the protein coat, they noticed that the radioactivity was found in the pellet, which implies that the DNA entered inside of the bacteria, and this is the one that would later on produce more viruses. So this would give the conclusion that it is the DNA that gives the set of instructions to produce viruses. Therefore, DNA is the genetic material. So all of this stuff was able to make sure or made us understand that the DNA is indeed